YouTube. This today is Dr. Mark Kahn and Dr. Doug Marchuk, and I'm going to read their introductions because I can't remember all of it. Dr. Mark Kahn is a cardiologist and the Edward S. Cooper, Norman Roosevelt, and Elizabeth Meriwether McClure Professor of Medicine at the University of Pennsylvania. As one of its many projects, his lab has been working on signaling pathways involved in CCM development for many years. Most of you are already familiar with his team's microbiome and emulsifier findings, but there is so much more, including what he's going to be sharing today. Welcome, Dr. Kahn. And we have Dr. Doug Marchuk. He's the James B. Duke Professor of Molecular Genetics and Microbiology at Duke University. Dr. Marchuk was one of our very first scientific advisors, and we've known each other for almost 20 years now. He has been at the forefront of vascular biology research, including CCM genetics and gene discovery, for as long as I've known him. He's incredibly supportive of patients and patient organizations, and we appreciate him. Welcome, Doug.
Thank you.
Dr. Kahn and Dr. Marchuk are collaborators on an NIH program project grant that also includes the University of Chicago and UCSD, and let me know if I've left anyone else out. This is the first of a two-part series on PIK3CA mutations. The second part will be on June 16th and will feature Dr. Issam Awad, and he's going to be speaking more directly about the clinical implications, including information about rapamycin and CCM. And then the last thing I want to say before Dr. Kahn starts is please ask your questions in the Q&A. Use the little Q&A button there to ask so that it's easier for us to keep track of them rather than asking them in the chat. Take it away.
Okay, thanks, Connie. As Doug said just before we started, this is already old news. It's been out for a couple of weeks. No doubt there will be a few folks who will ask why we're not showing any unpublished data. But anyway, let's start. So the story, as Connie suggested, is a recent one that Doug and I and our labs have kind of teased apart, showing a role for PI3 kinase mutations and PIK3CA mutations in CCM formation. There we go. Okay, just quickly, CCMs are, rather common. If you look at autopsies, we know that they form in the postcapillary venules of the central nervous system, a common cause of neurologic deficits such as stroke and seizure, especially in young people. Management of CCMs, and then Issam will no doubt talk more about this, remains pretty simple. It's mostly conservative, with the exception of neurosurgical resection of reachable lesions. And this is just an MRI of a patient with a familial CCM disease who has a large number of lesions. And many of you are already familiar with this, so we'll go through it quickly. The disease has been known by clinicians to be broken up into a familial form of the disease that is autosomally dominant in its inheritance pattern, and the more common sporadic form. The familial form enabled folks like Doug, human geneticists, to clone the genes that cause the disease, and presents earlier than the sporadic form, and of course with multiple lesions. And the genes that were identified to date have been three genes that encode three non-homologous proteins. CCM 1, 2, and 3. They form a fairly stable complex, although the CCM3 protein is less stable than CCM 1 and 2. And we know that a loss of any one of these three genes will disable the complex and result in a loss of CCM function. And this is the sort of underlying mechanism for much of CCM disease. Familial patients have a germline mutation in one of these genes. And then only until our recent study, we thought that all they really needed to get CCMs was mutation in a second allele. And that may still be true for many smaller lesions, but as I'll tell you, we're now up to something like a 3-hit hypothesis on top of the 2-hit hypothesis for more aggressive lesions. Okay, so as Connie mentioned, my lab has worked on the signaling aspect of this disease for some time. And probably the most important thing that we found is that the real role of this complex is to bind and negatively regulate the function of a MAP kinase, MEKK3, that is particularly important in endothelial cells. And that MAP kinase will activate a cascade that ultimately results in the expression of two homologous transcription factors, KLF2 and KLF4, in endothelial cells. And it's when you have an excess of KLF2 and 4 that lesion formation takes place. And we've shown previously that you can actually reverse lesion formation by reducing the input of MEKK3 or KLF2 or KLF4, at least in mice. And this is a picture of a mouse hindbrain that has CCM lesions after loss of the CCM1 or KRIT1 gene with an endothelial deleter. Okay, so the story sort of begins with a fact that we've known for a long time since Elizabeth Tournier-Lesserve published an elegant paper with Bouldet as the first author, demonstrating a mouse model of disease. And what her lab demonstrated, and many others such as ours has shown since, is that if you delete any one of the CCM genes in the very early perinatal period, you will get CCM formation, not everywhere, but in the retina and in the hindbrain. And these are just pictures, visually and microscopically of these early lesions. But if you delete later in life, for example, in this experiment at 2 months, what we and others have found, and the Tournier-Lesserve lab also published a paper on this quite a long time ago now, is that you no longer see lesions in the brain. And this is just the CCM knockout brain compared to the control brain. Now you see there are no lesions formed. But when we followed these animals carefully for 4 months, we did find a single place where there was lesion formation. Even though the brain was negative, the testes in these animals formed very large lesions. And I won't show you all the details, but histologically and molecularly, these lesions really are cavernomas. They're just testicular cavernomas, not cerebral cavernomas. And so this was an important clue, because it suggested that you could form cavernomas in older animals, but you weren't forming them in the brain. And since we know that most of these cavernomas arise in adults, in humans, the question was, why weren't we seeing them form in older mice? And when we took a look at the endothelial cells in these older mice after having deleted the CCM1 or KRIT1, we could see that we were indeed inducing a high level of KLF4. We have a good antibody for that nuclear transcription factor, and you can see the red is the endothelium, and the green here is the nuclear KLF4. So we were having the predicted signaling effect that we had shown to be causal, and what we thought was sufficient for CCM formation in the neonatal model. But in an adult animal, it was no longer sufficient, and we couldn't get lesions. So the question was, why not? We began to read about this, and one thing that stood out was, this is a study that was done in rats quite a long time ago, almost 20 years ago. And what this study did was just look at endothelial cell populations in the various different organs in rats, and using BRDU labeling, asked which ones have higher and lower proliferation. And what the study showed was that the testis stood out because it had the highest level of basal proliferation in the adult animal in the endothelium. And the brain stood out because it had one of the lowest. And so that was kind of interesting. And it suggested that, when you look here, you're looking at really like an order of magnitude difference, that maybe there was a signal for proliferation that was required for lesion formation and that signal was present in the neonate, especially in areas that are still angiogenic after birth, such as the hindbrain and the retina, but that it was absent in the adult, and that something else had to happen to provide that signal. And indeed, as I just mentioned, if you look in the neonate at P1, you can see large CCM formation here in the retina. If you do it 3 weeks, when the retina is no longer actively growing, at 2 months rather, you'll see that there is no lesion formation. Okay, so We looked at the literature, and one thing that also that struck us was that, there was a large role for PI3 kinase gain of function mutations. PIK3CA is the enzymatic component of the PI3 kinase enzyme, and gain of function mutations in PIK3CA have been found to cause both venous and lymphatic malformations. We knew that CCM lesions were pretty much post-capillary venous lesions, and therefore fell into this category of venous/lymphatic malformations. And so we began to wonder if there was a role for proliferation in all of these different malformations, and that might also be intersecting with the pathogenesis of CCM lesions in adults where there was not enough basal proliferation to support lesion formation. And so what we used was a PIK3CA gain of function model that others have used, such as Michael Dellinger in Texas for lymphatic malformations. And we used a brain endothelial specific CRE. This was given to us by Marcus Schwaninger. And we used the same CRE to induce both loss of CCM formation with the CCM CRIT1 floxed allele, and to induce gain of PI3 kinase signaling with a PIK3CA gain of function allele that would drive expression of an H1047R mutation, which is a mutation commonly found in cancer and in other vascular malformations in humans. And what we identified, and when I say we, I mean Eileen Wren in the lab, what Eileen found was that there was a very strong synergy in the neonate between these two signaling changes. When she took, these are animals that are on what we would call a non-susceptible or resistant background. I won't go into the details, but it turns out that the MEKK3 pathway is strongly regulated by the microbiome, even in the brain endothelium. And when the animals have a resistant microbiome and we delete CCM1, we get little or no lesion formation. When we activate PI3 kinase, we do get lesion formation. But when we activate PI3 kinase and delete CCM1, we get a really tremendous synergy. And that is shown here in this sort of elegant lesion volume measurement that was done blinded by the Awad Laboratory in Chicago, whom we're lucky, fortunate enough to collaborate with. So this suggested that there was a strong synergy between these two pathways. And when we looked further at the types of lesions that you get with CCM loss of function and PIK3CA gain of function and PIK3CA gain of function and CCM loss of function, it was striking how similar these lesions were at the histologic level. And what I mean by that is that the CCM lesions in the susceptible background will form in this post-capillary venule in the white matter. And when we looked at the PI3 kinase gain of function, we saw very similar lesions forming in almost the exact same place. And the synergy, of course, arose in that place as well. So there was sort of a common denominator with regard to the types of lesions that were forming with all of these genetic changes. Okay, so what happens in mature mice? Well, we tried to do this experiment initially by simply activating the CRE using tamoxifen in adulthood. And what we found was that the animals that carried both a floxed CCM1 allele and the PIK3CA gain of function allele with the CRE would basically die by adulthood even without tamoxifen induction. And this turned out to be due to leak of the CRE transgene in the absence of tamoxifen. And the leak, when it occurs in places like the spine, result in massive neurologic, symptoms and actual death. So literally, we had no animals to assay in adulthood. So we developed a different approach, and this was developed by Sophie Sue, a very talented neurosurgeon who joined the laboratory for a year, was very productive. And what Sophie did was create really what is a cranial window. Basically, we made a small hole in the skull of these animals. And rather than using a genetic system to deliver CRE, which we had found to be very imperfect because of leak of that system, we were now going to deliver the CRE by injection of a viral vector. And so this would get around the leak. And indeed, when we do that in animals that have both conditional CCM1 and gain of PI3 kinase alleles, we find that we can induce a very robust lesion formation. And this is post-op day 7 through 21. What we're looking at here is this cranial window that's been made in the side of the skull of the mouse. And we're looking at 7 to 21 days after we inject the CRE virus in with a needle. And what you see is not much happens for about 10 days. By 14 days, we can see really what is the tip of an iceberg of a large, massive lesion. And by 21 days, you can even sort of appreciate the sort of very classic mulberry-like appearance that the clinical folks and the surgeons have often described these lesions having when they are approached neurosurgically. So indeed, when we combine these together, what we find is that adult animals that would not get lesions with CCM1 loss alone do get lesions when there's a combination of CCM1 loss and PI3 kinase gain. And this shows you that not only does that happen, but that is actually required in the mouse model. These are animals with only CCM1 loss of function, only PI3 kinase gain of function, and both. And you can see this is just a sort of visual example of no lesion, and lesion. This is the micro CT, no lesion, lesion. But maybe most convincing, of course, are the data from Isam Awad's group, which actually looked at the entire brain and assessed in a blinded manner the volume of the lesions. And there's basically no lesion formation with either one of these mutations alone, and very robust lesion formation with both. Okay, we'll go to mechanism now, but I'm sure that you can see at this point, like, where Doug and Dan Snellings came in to look at, whether human lesions might also require this dual mechanism. And we'll come back to that in a few minutes. And then I'll just finish my part of the talk on the mechanism. So, we want to, synergy usually implies at the molecular level a point of intersection between two pathways, and like sort of two waves that, at the beach, one comes upon the other, and then you have a massive wave together. And so the first thing we looked at was whether PI3 kinase might actually be upstream of MEKK3 and provide an input into MEKK3. And that would, sort of give us a kind of super MEKK3 activation mechanism. And of course, we know that the mechanism by which MEKK3 functions in endothelial cells is through the KLF2 and 4 transcription factors. So we looked at KLF2 and 4 in the endothelium of animals that had loss of CCM only, gain of PI3 kinase only, or both. And what you can see here is, as before, we can see an upregulation of KLF4 with loss of CCM, but there's no upregulation of KLF4 in the PI3 kinase gain of function lesion and endothelium. Of course, it is present when both are there. So this suggested that PI3 kinase was not increasing KLF4 expression, and therefore it was unlikely to be increasing MEKK3 activity. And when we did a similar experiment using just harvested isolated endothelial cells from the hindbrain of neonatal animals that had either of these different mutations. And again, you can see an increase in KLF2 and KLF4 with loss of CCM1, but no change in KLF2 and KLF4 with a gain of PI3 kinase. And the same data can be reproduced actually entirely in vitro if one uses siRNAs to knock down CCM1 or lentiviral vectors to drive expression of the PIK3CA gain of function allele. So this, pretty convincing evidence to us that the mechanism was not by PI3 kinase feeding into the MEKK3 KLF24 pathway that we'd already figured out was important for disease mechanism. So then we wondered about whether it was the other way around. Is it possible that PI3 kinase is, the most important common effector pathway? Again, PI3 kinase plays this critical role in lymphatic and other venous malformations. And when we looked at the types of lesions that PI3 kinase can create in neonatal animals on its own, they really looked almost indistinguishable from CCMs. So here's a schematic. In this sort of schema, we're wondering whether the CCM loss of function and MEKK3 gain of function is synergizing with this pathway by merging with it and sort of adding to PI3 kinase. And it's difficult to measure this in vivo. What we're looking at here in green is phospho-S6, which is an effector of the mTORC1 downstream of PI3 kinase. And indeed, no surprise, when you look at the PI3 kinase gain of function animal, you can see high phospho-S6. But what was really striking to us and to Aileen was that when we looked at the CCM knockout, we also saw a very large increase in phospho-S6 in those endothelial cells. So this looked like this might be the point of synergy. And then when we add them together, there's extremely strong green staining. So it looked to us as though MEKK3 was in some manner upstream of PI3 kinase, or at least upstream of mTORC1 in the PI3 kinase pathway. And this has been confirmed by both analysis of endothelial cells harvested from genetically mutant mice and from, again, the in vitro system. These are endothelial cells taken from the hindbrain of CCM1 endothelial knockout animals. And you can see these are Western blots that were done after magnetic sorting of these cells. And the phospho-S6 is subtly but significantly raised. We couldn't see a change in phospho-AKT in this system. And again, with HUVEX, we see a very similar set of data. Maybe it's a little more robust because of the system and the fact that it's easier to isolate these cells and lyse them quickly. But you can see an increase in phospho-S6 with loss of CCM function. And so this, one last tool we had at hand was, for years we'd known that KLF2 and KLF4 are critical effectors. So we developed the KLF4 gain of function system. This is a TET-O KLF4 allele that is activated by doxycycline. If we combine a number of different alleles, we can drive it specifically in the endothelium of the brain, which is what we did. And when we do that, we could see a pretty significant lesion formation with just KLF4, again, consistent with sufficiency of KLF4. And when we look at this important signaling readout, we find that KLF4 gain of function raises phospho-S6, and more evidence that the MEKK3 KLF pathway is proximal and driving PI3 kinase, or at least mTORC signaling. And again, if you look at the lesions that you get from KLF4 gain of function, you have that classic white matter venous or a capillary to venule lesion formation. This is just a little more data from that was generously worked on with us by Marco Castro and Michael Potente's lab. And Marco used lentiviral vectors to induce KLF4 in HUVEX. Again, showing a very robust increase in phospho-S6. And in this, I would say maybe more sensitive system, he was also able to show an increase in phospho-AKT, suggesting, again, not proof, but suggesting that it's activating the entire PI3 kinase pathway. And so, sort of to end what our part of the story, we think that the genetic and the molecular and biochemical data point to an important synergy between loss of CCM function, gain of PI3 kinase function. And this synergy also tells us more about how loss of CCM function is giving us these lesions. And we believe it's actually, to a large extent, feeding through PI3 kinase, the PI3 kinase pathway, and that there is a still undefined mechanism or mechanisms by which gain of MEKK3 and KLF24 are augmenting PI3 kinase, AKT, and mTORC1 activity. And I'll finish with just a little bit of a teaser for translational work. We know that, as I showed you, mTORC1 is the critical effector that we've been able to see with phospho-S6 activated with all of these genetic changes. And rapamycin is a well-described and FDA-approved antagonist of mTORC1. So what happens if you give rapamycin? If you give rapamycin to the neonatal model, you get a very dramatic reduction in CCM formation. If you give rapamycin to the adult model that Sophie developed, where you have really a robust lesion formation and developed requiring both CCM loss of function and PI3 kinase gain of function, get an even more dramatic change. where even low-dose rapamycin can shut down lesion formation, and high-dose rapamycin shuts it completely. And rapamycin also has an effect on the KLF4 gain-of-function model, although less of one, again, proving that the mechanism by which these lesions are forming is at least in part through activation of that pathway. So the summary for our part of the talk is that PI3 kinase signaling and CCM loss of function synergize in the neonatal mouse model. The synergy is required for the development of lesions in the adult. Either one alone is not sufficient in an adult animal. We can go into why that is if people have questions. The next part is Doug, so I'll tell you a little bit about that. This is, sorry, I should have edited these slides a bit more carefully. And we think that PI3 kinase signaling is downstream of CCM loss and gain of KLF function. And then I guess what we're all interested in doing now is really taking a closer look at the PI3 kinase pathway at FDA-approved drugs that have been developed mostly with the context, the goal of treating cancer in mind, and what they might do in a CCM context. And the folks in my lab who did all the work, Eileen Wren was the primary person. As I told you, Sophie developed the adult cranial window assay. Courtney Hong and Alan Tang really contributed an enormous amount of a number of experiments. Alan actually started the whole experiment. And we've had great collaboration, of course, with Doug and Dan and Isam's group and the folks like Connie at Angioma Alliance. So now I'll hand it over to Doug and he'll tell you about the human data. I'm going to stop sharing, Doug.
Right, okay. I will, oh wait, I'm not on my first slide. Go to the first slide. There we go. I hope you can see my screen and I will put it on projection mode. Here it goes. Yes, you can see it. Great. You can hear me? Great. Well, thanks, Mark, and especially thank you, Connie, for allowing us to share this exciting data. And again, I'm going to be talking about the human genetic and genomic studies of CCMs, taking off where Mark ended. And again, this work was done in my lab and done primarily by Dan. And we're going to talk a lot about the Chicago team's contribution in a minute. Just to remind you that the CCMs occur both as inherited and sporadic disease, and the familial forms are often shown by multiple lesions, whereas the sporadic usually have a single solitary lesion. And there are 3 known genes that can cause this. And what we had shown, actually Dave McDonald had shown, was that even in the sporadic cases, there are somatic mutations in these same genes. And so that led to a model of pathogenesis, and this is prior to Mark's mouse work, that we would say, well, if you were in a family, you already had one mutation that you inherited from one of your parents, and all the cells in the vasculature of the brain, showing a brain here, have one copy mutant and one normal. But that some point in time, and happening presumably multiple times over the course of the lifetime, that normal copy of the gene got mutated, and that seeded the formation of a CCM. And that's why there are multiple ones, because in a sporadic case, because you don't start out with one mutant copy, every cell has 2 good copies, so you need 2 mutations to occur in the cell. And that's very, very rare. And I always like to say, this is like the brakes in your car. You have the foot brake and the hand brake. And if one's already broken, you're halfway to disaster. If you have both working, you can still stop the car. Here, this is going to be a rare event, but it does happen. And when it happens only usually once. So when Mark told us about these early on, even in some of the studies that Eileen was doing, and showing that by activating PI3 kinase in the mouse, they could get this super phenotype. We said, well, let's go back and start to look at some of the CCMs we had been studying and see if we could find PI3 kinase mutations. And lo and behold, and this is the work of Dan Snelling's, he showed that although we have some CCM mutations in here, in many, and this is the color code, you can read it up here, but in many cases, we found these very specific PI3 kinase mutations. Mark had mentioned that he made a mouse with this particular mutation called H1047R, but we found that mutation in some of the CCMs. And we won't get into this, but the key point here was that if you look at all the mutations for PI3 kinase in cancer from a database called the Cosmic Database, the catalog of somatic mutations in cancer, and this bar graph shows that the longer the bar, the more common that mutation. If you look, the same common mutations that are found in cancer, we were finding in obviously a much smaller data set. This is the compilation of thousands of labs. This is our lab, but you can see that it's the same mutations. So these cancer-causing mutations are showing up in these CCMs somatically. That is, they're happening in the lesion, although the patients don't have them in their normal DNA and the rest of their body. And this made us think, you know, this is a kind of a paradigm of cancer, where cancers, they grow because they continue to get multiple hits in different genes representing different pathways, so that ultimately, and this is like the Vogelgram for colon cancer, but you've got the same idea that you start with normal cells and eventually you end up with a tumor. Now, are vascular malformations not in a cancer way, but are they following the same multiple gene mechanism? And I want to point out something, that this works because ultimately there are some of those cells that have all these mutations in the same cell, and that's the aggressive cancer. Is this really happening in the vascular malformations? So here is the summary at the beginning. So what we think is these vascular malformations have this cancer-like mutation mechanism where we often have to acquire mutations in growth suppressor genes, which we think of as the CCM genes, and growth activating genes like PI3 kinase. So let's go into the data to support that. Now, I already showed you that we found these PI3 kinase mutations in the CCMs that were surgically resected from patients, along with sometimes mutations in the CCM genes, which we already knew were there inherited, and we even knew that were acquired somatically. The question is this really like cancer? And if it was, they should occur, as it says, in the same cell. And this is just a picture of this when we're sequencing DNA from a big lesion and we just grind it up and make DNA. We can see these mutations, but we don't know what cell they came from. We need to really isolate individual cells to see that, like, for example, this blue cell doesn't have any mutations, but this orange cell has a mutation in PI3 kinase and in CCM, etc. So that's what we need to do. When it's all mixed up in a big gamish, we can't tell. So now I'm going to tell you a little odyssey of how this works, and this will be less science and more show and tell. But what we decided to do was single cell genomics. Now I want to point out something, that we obtained these CCMs in archive biobanks. So they're frozen, and one cannot isolate individual cells from a chunk of ice, but you can get the nuclei. They're a little bit more sort of hardy. So this just happened this morning. We got a new frozen CCM, and just this morning I took this picture on Dan's bench. Here's a little frozen CCM in a tube, and you can see the scale there. And I just want to remind you that the nucleus, and again, I'm gearing this toward perhaps the lay audience here, but in the DNA is in the nucleus, and it has a membrane, a nuclear membrane, and the rest of the cell is encapsulated in the cell membrane. This is extremely fragile, and if you get a frozen piece of tissue and you start to mush it up, it's gone. But the nucleus, with certain tricks, you can keep it intact, and that's what we're going to try to do. So we've getting these frozen nuclei, and we're going to get the frozen cells. We're going to get the nuclei out. Again, a photograph just this morning because we got a new frozen CCM, and Dan is prepping that frozen thing that was in the tube, that frozen CCM. He's lysing all the cells. The cells are popping open, but the nuclei, like a little kernel, is staying intact mostly. we could quibble about that. And then what do we do next? Well, and the next thing we have to do is we have to sort those nuclei away from the all sorts of other junk and debris. And so we do this by fluorescence activating cell sorting, or actually technically, because we were now working with nuclei, fluorescence activating cell sorting. This Dan took this picture this morning when he went to the fax machine and started to work on it. And as he pointed out to me this morning, fortunately for you guys, it clogged for a moment. So we had to take it, crack it open. And here's the inside of that thing. And it got unclogged, by the way. But you could see this plate here and a light, and it looks like some kind of laser beam. And what this, I want you to focus here on this inner part, because what it's really doing is it's shooting these cells, or in our case, nuclei, through a nozzle, and it's going to break the stream into very tiny droplets. And hopefully only one nucleus or cell, in our case, nucleus, is in each droplet. And then it's going to have a little charge system based on a dye that we put in the solution. And the nuclei are going to be deflected into one tube, and all the rest of the debris, cellular debris, is going to go in another tube. So we're going to purify the nuclei away from all this other cellular debris, which there's a lot of, by the way. And so we got that. And so we have a tube now, not of cells, but of individual nuclei. But remember I told you we wanted to study each nucleus separately. So how do we do that? We need to identify the DNA from each nucleus. So we're using something called the Tapestry platform from Mission Bio. We're not getting a discount yet, even though we keep advertising for them. And this is going to use this instrument here is going to use microfluidics to isolate each of those nuclei Dan now has in the tube into a tiny micro droplet. So here's a picture of kind of the workflow. You're going to have these all these nuclei, and they're going to be encapsulated in a little oil droplet. And then we're going to add chemicals into that system where we to pop open. Now we're going to pop open that kernel of where the DNA is. And we're going to do this thing called barcoding, where each nucleus is going to get a series of little snippets of DNA that will be amplified with the DNA that's in that single nucleus, so that at the end, those are coded with a little sequence code, so we know what nucleus it came from. And then we're going to break it back open into one big gamish and amplify everything. But now each individual nucleus has been encoded, or barcoded, as we say, so that we know all these sequence reads came from one nucleus. So then this is my little picture of microdroplets with sample going in and being popped into a microdroplet. So this is obviously blown up very, very high scale. These are very, very tiny droplets. So if you look under the microscope, and we did a little bit of this at the front end to see if we could make nuclei, you see all these dots. Those are the light color blue dots are the microdroplets. And often you see a single nucleus in many of these. This is the bright blue, and this is a stain for the nuclei. And most of them contain only one, and a lot of them don't contain any nucleus, and very few contain two or more. So this is the way this works: is you're going to make sure that almost always, if you get a nucleus, you only get one. That means sometimes a lot of them don't get any. Okay. Anyway, so then what do you do next? Well, we take all that after they're barcoded. We could put it back together and take all that DNA and sequence like crazy. And so I'm saying deep DNA sequencing. We're trying to get about 2,000 sequencing runs per gene that we amplify in there, per nucleus. So we're trying to get enough sequence data that we can really know what's going on. And here's one of the few pictures I'm going to show from the paper. And basically what we found was these individual nuclei, when we sequence them, if we could find the mutation, it's always, in this case, all 3 mutations in the same nucleus. And here's a couple cases. And this is highly, highly significant. We did it on 5 samples, some of which were from familial cases, some of which were from sporadic cases. But in all cases, there were nuclei that had, in these 5 cases, that had all 3 mutations in the same nucleus, meaning in the same cell. And this is fulfilling that kind of cancer-like paradigm. You need multiple hits in the same cell to make that really begin the clonal expansion of a very aggressive cell. So here's a modified model. I showed you this. This is a picture that Dan had made, and then we just added to it. We're saying that in a familial case, well, you've already inherited a mutant copy. You might get a somatic mutation and knock out that second copy. But then eventually, if, and here's the point, if you get a somatic CCM mutation, you get perhaps a highly aggressive CCM. And what we're wondering is if there's a bias here in the sense that the lesions that we get from surgical resection, even if a patient has 50 or 100 of them, the one we get is the aggressive one. And that's why there's such a high incidence of these PI3 kinase mutations in those, because maybe 70% of in our series, because those are the aggressive ones. Now, in a sporadic case, and I won't get into the details because we have, and Mark talked about unpublished data. Dan has some really good unpublished data to help us understand this, but we think a somatic PIK mutation may cause the propensity to get a CCM, and then by just causing perhaps a DVA or something, and then so the PIK mutation may come first, and then 2 somatic mutations. So anyways, I want to also acknowledge the people who worked on this. And again, Dan did almost all of this work with Matt and Chris and Mackenzie and Carol. As always, here's our lab. And again, this is a collaboration between Mark Kahn's lab and Dr. Awad's lab and his team. And it was a great collaboration. And I want to acknowledge especially, again, the funding of the NIH and the support of Angiome Alliance with Connie and Amy. And where am I stop sharing? Did I do it? Yes. Okay. So we do have some time for questions. Good.
Yes, we do. Thank you. I have the first question, and it's not in the Q&A. So it's
Okay.
So the work that was done previously with emulsifiers with the microbiome, that from what I heard was potentially the third hit. How does that relate to these new findings, or does it not?
I guess I'll take that one, Doug.
Yeah, go ahead.
Yeah, so the way to think about it, I think, Connie, is that the microbiome regulates the input to MEKK3. And most of that being, at least in the mouse, through TLR4, but probably other innate immune receptors as well. And so what happens is, you have basically 2 different pathways that are being disrupted. One, the CCM pathway, and the other, the PI3 kinase pathway. The CCM pathway is really there to modulate MEKK3. But, going back to sort of Doug's, brake analogy, hand brake and foot brake, there's also an element of the gas in this thing. The microbiome determines how much gas is going into the engine. Maybe you don't need the brakes if there's no gas, right? And that's why we can get animals that we take away the CCM function, even in the neonatal state, where we know that's almost always sufficient to give a lesion, but we don't get any lesion formation. And that's because their microbiome is such that the gas going into MEKK3 is pretty little. And MEKK3 signaling is kind of naturally downregulated in that setting. Having said that, one thing we didn't talk about today, and Doug is more expert in this than I, but there have been, because he knows these papers very well, there have been a number of other labs, and Doug's lab has confirmed this, that have identified gain of function mutations in MEKK3 itself being sufficient to cause, CCM formation. And so, that would be kind of like a microbiome independent. We predict that would be more microbiome independent, because it short circuits the microbiome, which is above MEKK3. So I would put the microbiome as a modulatory mechanism, not an essential mechanism necessarily. It's a very interesting question whether lesions that have both PI3 kinase and CCM mutations are also microbiome influenced to the same extent as the ones that we identified that were, really modeled on CCM loss of function only. And that's, I think, a big question. And as Doug said, we are not looking at all the lesions in a person, in people. We're looking at the ones that go to surgery, the ones that are really bad and growing quickly, maybe bleeding, and are kind of pushing the patient and the doctors to go to surgery. So these are open questions, but, and it may be that in those lesions, microbiome doesn't play that big a role.
Okay, thank you. All right, so that transitions over to a question for Doug. What is your opinion on MAP3K3 or MEKK3 mutations found in the two papers by our Chinese colleagues, Hong et al. And Wang et al.?
Yes, I mean, that was beautiful work, beautiful work. And just like Mark said, we have in fact confirmed that. It's interesting, Dan sequenced not just the CCM genes and not just PI3 kinase, but in various different panels, a whole host of different genes. And because MEKK3 isn't listed in cosmic, any event, we didn't put that on the panel, or it wasn't on existing panels, and we didn't augment it. Huge mistake by my part, I'll take the blame. So we didn't notice that, but yes, we found that, and it's absolutely true. And if you read both papers and sort of read between the lines a little bit, you can see that they seem to be, like if you don't have a CCM, well, I won't get into it, but it seems like either the CCM pathway is either activated by inactivating CCM or by activating MEKK3. I should say that that pathway is activated by either knocking out the CCM pathway or activating CCM3. Absolutely there, absolutely strong data, correct. And yes, and they are in sporadic lesions because the other ones have CCM pathway genes knocked out.
Mark, the next question is for you, I think. Were you able to determine the putative cell of origin of your cavernomas in your AAV-CRE model?
Yeah, that's a good question. The virus that we used was pretty non-specific. We know that it infects, cells that are non-endothelial cells. We feel that a lot of the work that we've done previously and others have done previously using different, CRE lines strongly support an endothelial cell autonomous mechanism for this disease. But our experiment doesn't address that. So, the answer is in that model, I'm assuming that endothelial cells are getting hit along with other cell types. But I can't tell you from that experiment that they are the causal cell.
Okay.
There's
Next
a lot of
question.
questions in the Q&A.
There are. Doug, I was wondering how you deduced that PIK3CA mutations occur prior to 2 CCM mutations in sporadic disease. I can understand that you can infer this from the frequency of mutations. However, tumors evolve over time, and you are assessing the current state of the tumor.
Yeah.
How do you account for this?
Yeah, I know. One way is, as you point out, you could do that by the frequency of mutations in the lesion. And I'll say, I probably, there's some unpublished data that we have that suggests that they happen first. I don't think we have proof of that. So I think it'd be better if I left it off until we have more data. But I think we can see evidence of that. But it's hard to tell in the frequency data because it depends on where you chop in the lesion. So I appreciate the fact that I didn't give any evidence of that, and I don't think we have enough evidence to prove that. But that's our inkling. Can I touch another question that Jun Zhang asked, which is a really common question that we always get, and that is that oh, maybe there's DNA instability in your, which happens in cancers, that one of the first pathways you knock out is the DNA repair pathways, so that then somatic mutations happen like crazy. But we've done a lot of calculations, and that is just not necessary to invoke that. And although it'd be really hard to prove that it's not true, we don't have any evidence that it's true because Dan never just sequences one gene. And we just don't see somatic mutation. Like if we sequence 16 genes or 20 genes or whatever genes we see, we only see somatic mutations in the one gene. And if these were happening by chance, we would see an increased somatic mutation rate. We don't have genome-wide evidence, but there's just no evidence that it's true. And if you calculate the number of endothelial cells just in the brain alone, and with some back of the envelope calculations, it's not that infrequent that you'd be incurring these somatic mutations, especially loss of function mutations.
Okay. I'm going to ask one of our patients is asking, so radiation is known to cause the development of more cavernous malformations, specifically if it's given for brain cancer, but we also wonder in the hereditary form whether that's true with gamma knife. How do you see this related to radiation being an instigating factor for lesion formation? I'm opening it up for both of you. I can see both of you don't want to answer that.
I don't think we really know the answer to that one, Connie. On one side, sure, radiation can induce genetic mutation. On the other side, radiated cells have a hard time proliferating and are more likely to become apoptotic when they try to proliferate. And so that would, biologically, we know that proliferation is a key step in this process, and so that might be protective. So the answer is we have no idea. I think that just everything would be speculation.
Okay. Next question. In the work of Detter et al. And the EU labs, cavernoma formation took place in 2 steps: clonal expansion and incorporation. Is the PIK3CA required for either of those?
So both of those models were in mouse. And we have now, Mark induced PI3 kinase activating mutation through a sort of a genetic trick. But in both the 2 papers that you refer to, there was no overt PI3 kinase mutation delivered, and I doubt whether there was a gain of function mutation that occurred in those lesions. So I don't, PI3 kinase may have a role, but I think that's much later than what those lesions were showing. In other words, it may have a role in further growth and aggressiveness, but that's not necessary to invoke that, because that didn't, almost certainly didn't happen in either. That would have had to have happened in the endogenous mouse locus. And there's no.
I think just to go back to the models again, Connie, I think one thing we've learned from these studies is, and this is what actually initiated these studies, every model has constraints.
Yeah.
And introduces artifact and fallacy based on the way it's designed. It turns out that the neonatal model was a great model. It showed us a lot of essential signaling pathways, but the fact that it takes advantage of a high level of endogenous proliferation is definitely not what happens in the vast majority of human CCMs. So I think some of the conclusions that we've drawn from the neonatal model will have to be re-examined in this adult model. For example, the microbiome, we talked a lot about that. It's possible that the microbiome has a very powerful effect in the neonatal model, but we haven't yet tested it in the adult model. It's one thing we need to do. It may not be as important, and also not as important in this kind of multiple gene mechanism. So I just, you learn more as time goes on, and conclusions need to be tempered as time goes on.
All right, next question. We still have time for a few more. The question is about hemorrhage. So the information that you've given us so far, as I understand it, is mostly about lesion growth. And is there anything to be said about hemorrhage from the things that you found? What triggers a bleed? What stops a bleed? What makes the lesions unstable, stable? Anything? I love that there's 2 of you, and
Probably not. I mean, well, Mark Ginsburg might have more to say on hemorrhage than we do. His laboratory, he and Miguel have worked a lot on this. Certainly, we know that the MEKK3 KLF24 pathway regulates endothelial, modulators of clotting, like thrombomodulin, in a very strong way. But again, I don't think there's anything that we've learned here that will speak directly to hemorrhage. One thing that might be valuable will be to look at this adult model specifically with regard to hemorrhage over time, because we think the adult model is a more accurate representation of human lesions, and we haven't done that yet, Connie. So we may get insight there, but we don't have it now.
Yeah.
Okay.
We got a lot of.
Next question is, were you able to demonstrate in your single cell analysis that both mutations occurred in an endothelial cell?
Yeah, so that's a really good question. Thank you for it. And in fact, as you will know, the person who answered this will know very clearly that the data so far is really strong, that from the mouse, from the human in the past, that these mutations are, the CCM mutations are occurring in the endothelial cells. When we did the gamish, we tried to use a endothelial nucleus marker to sort, and we had some difficulty getting it to work. So we just isolated the nuclei. Having said that, because the same mutations are known to be in endothelial cells for the CCM, I think it's highly unlikely then, and we found the PI3 kinase mutations in the ones with the CCM mutations. I think, it's just obvious that they are in ECs, but we were not able to sort endothelial nuclei, at least at that time. I know that there's a marker one, and we tried it, and we needed to get to work, so we just did the isolation without the nuclear antibody. Good question, though. Excellent question, actually.
You're muted, Connie.
The rest are comments. Jana Burkholz says, that's amazing. This is to Doug. Thank you so much for showing us how you sequence a single nucleus. That is so fascinating. And there were a few other thank yous in here. Do you all have anything else that you would like to share before we close?
No, I just once again, I would say this, that this work illustrates a couple things. One, the importance of collaboration across a number of different groups. It also shows the importance of the patient advocacy groups, because so many of these lesions either came from Dr. Awad or from Angioma Alliance. And and not just the lesions in this paper, but the CCMs, I'm saying, but the surgically resected CCMs either came from the Angioma Alliance Biobank or others. So the cooperation of the scientists, but also the patient advocacy, the patients in the patient advocacy group, this is how you get things done.
Thank you.
That's what I would say.
Well said.
All right. Well, we are done then. Thank you everyone for attending. I hope that you learned as much as I did. And we will be reconvening again June 16th at 7 p.m. Eastern time to hear from Dr. Awad about rapamycin more specifically, and also updates on atorvastatin and COVID and biomarkers and lots of other stuff. He always has a lot to say. So thank you and have a wonderful rest of your afternoon.
Thank you, everyone. Bye-bye.